This is a 0 p.m. webinar. For those of you who don't know me, I'm Sarah Hale and I work for the German Water Centre. And for those of you who don't know 0 p.m., it's a Horizon 2020 research and innovation project where we're looking at interlinking prevention, prioritisation, and removal strategies to protect to protect human health and the environment from these persistent and mobile substances. It's a bit of a mouthful, but that's what we're doing in the project. Today, it's my great pleasure to welcome Daniel Zahn, and he's going to give the webinar today, which is called Chemicals Without Borders, PMT vPvM Chemicals from Release to Human Exposure. So I've known Daniel for a very long time. He's an environmental and analytical chemist. He has his PhD from Leipzig in Germany. He's still working in Germany, and his PhD was part of the Promote project, which is actually where I met Daniel. He's been looking at PMT and vPvM substances basically since as long as I can remember. And at the moment, he works at UFZ. So, Daniel, I'm going to let you take it away and bring up your slides.
My presentation today is named Chemicals Without Borders. So I want to have a look with you how PMT chemicals spread in the water cycle. If we have a look at the water cycle, we often talk about a multi-barrier system because we want the water cycle not to become a chemical cycle. So we have our wastewater treatment plants where we can have biological degradation, chemical degradation if there's advanced treatment, and sorption processes taking place. It is similar in surface water. We can have biological degradation. We can have sorption to soil and suspended matter. And in the top layers, we can have photodegradation. Then when we have the transfer to groundwater during infiltration processes, again, there could be biological degradation, this time in the soil, sorption to the soil or reaction with mineral surfaces. And in the end, when we treat our water for drinking water production, it's again the same similar processes here. We have sometimes biological degradation, sorption, and chemical degradation. So what does this in the end mean? We have three processes that dominate our water cycle, that dominate the barriers in our water cycle. First of all, we have dilution. So we put a little wastewater in much more clean water. And in the past, dilution often took care of things, but it becomes less and less reliable because, especially in hot summer months, we have small rivers that are up to 50% and more wastewater during the summer. And with the increased water needs, we have to decrease the distance between wastewater and reused water, either indirectly or in some countries that are really dry. There's also direct wastewater reuse for agriculture and sometimes even for drinking water production. So you see, we can't rely on dilution that much anymore. So we need to put more emphasis on the barriers we put in the water cycle. And if you have a closer look, all those processes here seem to depend on two mechanisms. On the one hand, transformation, which doesn't work too effectively for persistent chemicals. And on the other hand, sorption processes, which are often not too effective for mobile chemicals. So PM chemicals combine all the qualities to break through these barriers and therefore spread quickly in the water cycle. Why is this important? The chemicals that are released, of course, impact the ecosystem. They can interact with all kinds of aquatic organisms. And if they can break through the barriers we use, for example, for drinking water production, if they can accumulate in plants, there's also potential for human exposure. So we need to know which chemicals break through these barriers, which one are present in high concentrations, to prioritize them for sophisticated risk assessment and to implement effective countermeasures if it's required. And therefore, I want to take you on a bit of a journey today. I want to follow the water. We will go from release to possible roads of human exposure. So we'll start at the wastewater treatment plant, have a look at bank filtration, have a look into tap water, and finally find out what happens if you irrigate with water contaminated with PM chemicals. Let's start at the source and the wastewater treatment plant, which is the main entrance pathway for many PM chemicals into the water cycle. What I want to discuss with you here is a study we did in two wastewater treatment plants. One of them using powder activated carbon and the other one using ozone followed by sand filters. We looked for over 100 PM chemicals in there and wanted to answer three basic questions. First of all, we wanted to know which PM chemicals are actually present in the biological effluent. Then how effective are those advanced treatment technologies in the removal of PM chemicals? And finally, which chemicals remain and are released in high concentrations into receiving waters? This is an overview of the biological effluent of those two wastewater treatment plants. Here are only shown the chemicals with concentrations greater 0.1 microgram per liter. In general, of the 111 chemicals, we detected 72 more than once, and 47 were present in both wastewater treatment plants. Among the highest concentration chemicals, those with concentrations higher than 1 microgram per liter, were of course some of the well-known chemicals like benzotriazole, guanylurea, valsartanic acid, thiotriazoic acid, but also some less studied or sometimes even novel chemicals, like these shown here. Very interesting about them are the two inorganic ones, the PF6 and BF4. We found them only a couple years early. We found them in 2001, 2021 for the first time in the environment. So what happens to this chemical? First, what's the summary? We found 72 of the 111, which resulted in a total PM burden of 100 micrograms per liter and 40 micrograms per liter, depending on the wastewater treatment plant. So there are a lot of PM chemicals present in the secondary effluent. Next, we had a look at the treatment technologies. We only had to look at here at those PM chemicals in concentrations greater 0.1 microgram per liter. And here you see a breakdown of the removal with powder activated carbon and with ozonation followed by rapid filtration through a sand filter. The powder activated carbon had a pretty even 1/3, 1/3, 1/3 split. 1/3 of those chemicals were removed well, so greater than 70%. 1/3 was removed partially, so between 30 and 70%. And for 1/3, there was no removal. While for the ozonation, we see there is more chemicals where we have good removal, almost half of them, but a similar amount of chemicals where there was more removal, and we found some chemicals where we actually observed formation, because this ozonation can lead to the formation of transformation products. On first glance, that doesn't look too bad, but if you have a look at total concentrations again, the powder activated carbon only reduced the PM load from 103 to 87 micrograms per liter. So while 1/3 of chemicals was good and 1/3 was partially removed, the total PM burden was only reduced by like 10%. The reason for this is that many of the high concentration chemicals we found were not really affected by powder activated carbon. It mostly removed the low concentration chemicals. The picture for ozone is a bit better. We reduced the PM burden by almost half to 19 microgram per liter, but still we observe more formation. So both technologies only lead to a partial reduction of the PM curtain in the wastewater, at most by half. We wanted to have a look if these technologies are complementary. So you see the removal during ozonation plotted against the removal with activated carbon. And you can see there are some chemicals for which both methods are effective, some chemicals for which neither method is effective, and then a range of chemicals for which there is a complementary behavior of both methods. So we had a closer look at this. And the wastewater treatment plant with the ozone actually had a pilot plant connected to it where there was activated carbon filtration after the ozonation. If we apply this, we see the synergistic effect. Suddenly we remove 60% of the chemicals well, and some of the chemicals that are formed are also reduced. But we have this very recalcitrant core of like 9 chemicals that is not removed by either technology nor the combination of both. So how does this translate to concentrations? Even when we combine both technologies, The reduction in the PM burden is fairly low still. We see, oh, now I can't see my screen anymore, which is a bit annoying. I did something wrong. Okay, I can see my screen again. We see even if we combine both technologies, we only go down to 13 microgram per liter. So 1/3 of PM chemicals remain, even which when we use what is the most advanced setup that's commonly used in wastewater treatment. So even this technologically sophisticated method is not an effective barrier against PM chemicals. Which are the chemicals that remain and how do the chemicals behave that we have here? Here, our rather novel or not too well investigated high concentration chemicals. And this is how they behave during the treatment. Some are removed by one of the techniques, some are removed by both, but some are removed by neither. And all these chemicals had concentrations higher 1 microgram per liter in the effluent, and we can't really do anything against it. What I want to point out a bit, you see those two inorganic ions. They are often used as anions for so-called ionic liquids, which are organic salts. They usually have either one or two organic ions combined. Here it's an inorganic ion that's often combined with an organic cation. And they're a bit designer chemicals because they can be tailored to many, many applications. We had a look at more of these ionic liquid ions. The other ones did not reach these high concentrations with greater 1 microgram per liter. But we found one of them, this NTF2, which is a novel PFAS that we also detected only in 2021 for the first time, could also not be removed. And there were 11 more of these ionic liquid ions not removed during advanced wastewater treatment. Let's sum this up. We discussed for the last 10 minutes about our different advanced wastewater treatment technologies and how effective they are or not. But the one important thing we have to keep in mind, they are usually only implemented for large wastewater treatment plants. So in many of the smaller wastewater treatment plants, these high secondary effluent concentrations that we saw will be directly discharged in receiving waters. The other thing that's important to learn here, while our advanced technologies are effective against some chemicals, they have systematic gaps, chemicals they cannot remove. And we need to know which chemicals slip through our treatment to be able to, of course, evaluate if they are problematic and if yes, take appropriate action. After discussing the sources with which the chemicals can be released into the water, I want to have a look with you into surface water concentrations and bank filtration, which is often the first treatment step for drinking water production. The study I want to summarize here, we took a look into two river systems, the River Rhine and the River Mulde, and into a bank filtration site located at the River Mulde. These are the chemical concentrations we found in our two rivers. We have a frequency of detection, so how often did we detect them? And we have a median concentration. Of our 127 chemicals we looked for, we detect 92. So many of the chemicals we were looking for were actually present. However, only 44 of them had a frequency of detection greater than 50%. So more than half are actually present fairly sporadically. However, there were two chemicals we detected in really high concentrations, up to 2.5 microgram per liter, not as maximum concentrations, but at median concentrations in surface water. Those were caprolactam and again, the BF4. Comparing our two rivers, we had 72 chemicals in both river systems, but we observed actually quite pronounced concentration differences. In general, the River Rhine was more contaminated. We observed higher concentrations there. When you look at this graph, you see basically the concentration ratio between the river Mulde and Rhine. So all the chemicals you have at the top going towards 10 or above the line had higher concentrations in the river Rhine, and all the chemicals at the bottom had higher concentrations in the river Mulde. We see we have a couple of chemicals which are in this intermediate range, but almost half of our chemicals were either much more present in the river Rhine or much more present in the river Mulde, showing that there can be pronounced local differences in concentrations of these chemicals. And there were a couple of chemicals that were only found in either of the two rivers. So what's the first thing we learned from this? In our surface waters, we found 92 PM chemicals, and we found some concentrations of up to 19.5 microgram per liter of these chemicals. So the PM burden in surface water can be quite high. And again, it was dominated by few high concentration chemicals. So these high concentration chemicals are exactly those we need to remove during drinking water treatment. And the first step to drinking water treatment is often bank filtration. So let's have a closer look at the bank filtration site located at the River Mulde. What you can see here are removal rates. Those are recorded over a time period of a couple months. Therefore, you can see some variations, especially for those chemicals that are in the intermediate range of removal. Of course, we have dilution with groundwater, which makes it a bit hard to determine, but we took some tracer chemicals to account for this. So we have like half of the chemicals were fairly well removed during bank filtration. So again, bank filtration is an effective barrier against a number of these PM chemicals. However, the other half was only partially removed. Some were not removed at all. And for two, we even observed formation. Right? Having a closer look which chemicals were not removed, we see we have carbamazepine, we have the PF6 we discussed before in wastewater treatment, but most importantly, among those non-removed chemicals were again the caprolactine and the BF4, the two chemicals we found in the highest concentrations in surface water. So like in the wastewater treatment, we have pretty much a similar picture. The technology is effective against a number of chemicals, but among the ones that slip through are some of the highest concentration chemicals we observe. So what does this mean for concentrations and PM burden? We still find 40 PM chemicals in the bank filtrate in varying concentrations. But since some of the highest concentration chemicals slip through, we can only reduce the PM burden down to 8.4 micrograms per liter, a lot of which was caused by caprolactam and BF4 alone due to their high concentrations. So far we saw all the barriers we have are somewhat effective. They reduce the number of chemicals, they reduce the total concentrations, but we have this a very recalcitrant core of chemicals that seem to slip through. If we look at tap water, it is often produced either from bank filtrate, from groundwater, or sometimes even directly from surface water. And depending on the source water, you usually have different treatment technology afterwards. We wanted to know what are the PM concentrations in tap water. So we did a study collecting tap water from 11 countries and 3 continents. We had a total of 32 samples, mostly out of convenience. Many of them were collected from Germany with 15, but we also had Botswana, Czech Republic, Italy, Lebanon, the Netherlands, Norway, Portugal, Switzerland, Sweden, and even the US. Having a look at the raw concentrations and occurrence of these samples, you can see here, you can see marked with A is the concentration in microgram per liter. And this line at 0.1 microgram per liter is what's often used as the risk indicator value for chemicals for which there's not enough toxicological data. And we can do FOD, which stands for frequency of detection. The blue bar is the FOD lower than 0.1 microgram per liter, and the orange bar is higher than 0.1 microgram per liter. The first thing we see, we have two chemicals that exceed this 0.1 microgram per liter as median concentration whenever they're present. Those are, again, the B4 we heard before, and TFMSA, which is an ultra short chain PFAS you can see at the bottom. We have 7 chemicals that exceed a tap water concentration of 1 microgram per liter at least once in our data set. The highest one of those is the PF6, the other inorganic anion, where we found a maximum concentration of 3.6 microgram per liter in tap water. And to also have a look at a chemical that has been popping up again and again over the last years is this NTF2. It is present, rarely it's present in low concentration, but we also found it in tap water. So what kind of total burden does this reflect? If we look at total tap water concentrations, we are again lower than we were in surface water. We are lower than we were in bank filtrate. Here we have a tap water concentration of 0.1 microgram per liter up to 6 microgram per liter of the PM chemicals. We see there's quite a large variation. And even if you look into waters from the same country, like Germany, there's a huge variation between concentrations we observe, while others look very comparable if you look at Venice and Rome. So with this, we wanted to do some statistical analysis to find out if we can observe patterns. What you see here is a cluster analysis and the heat map. The heat map gives you indications about the concentration of chemicals in certain samples, and the cluster analysis is, well, in the end, clustering them together. We observed 2 different types of clusters. We observed the cluster for samples and the cluster and 2 clusters for chemicals. For the samples, we have this cluster samples 1, cluster samples 2. In this cluster samples 1, we found many chemicals on average, 20.5 plus minus 2. And in this cluster 2, we found few chemicals, 11.5 plus minus 2.2. If you have a look at the box plot down here, this is something I found quite fascinating. Of course, it's not surprising that we have chemical samples with more and with less chemicals. What was quite surprising to me is how clear this divide is. It was statistically very significant, with p below 0.0001, and the very clear divide of almost 10 chemicals in average on difference. So this was the first very surprising thing we observed. The first thing that comes to mind when you see this, that this could be a consequence of different treatment technologies. So we had a look at these samples, and we could not observe any pattern in terms of treatment technology used. So our working hypothesis right now is this is mostly connected to raw water quality, and indicates that raw water quality has actually a larger influence in treatment technologies, especially for those very hard to remove chemicals. Of course, this needs further confirmation. The second cluster we observed was a cluster with the chemicals. You again see on the top, this one's a bit harder to read, this cluster chemicals 1 and cluster chemicals 2 in the figure. This chemicals cluster 1 are chemicals with a high detection frequency. They were on average detected in 77 plus minus 15% of samples. So those are the fairly ubiquitous PM chemicals. And there was a cluster with a low detection frequency, with around 33% plus minus 16%. And here again, the 2 clusters are quite well separated, and it was statistically very significant. And I think it becomes very obvious also when you look into the heat map. What's quite interesting is when you compare both clusters for chemicals and for samples, The cluster 2 chemicals, so the rarely detected chemicals, are also the chemicals that you rarely find in the less polluted samples, while the cluster 1 chemicals are those that you find fairly often in the less polluted samples. Of course, the first thing that could come in mind is that you think, okay, those cluster 2 samples also have a much lower PM burden. However, this is not necessarily true because if you have a look at mean concentrations of the chemicals clusters 1 and 2, you see that on average they are not statistically significantly different, but many of our highest concentration chemicals are actually in this cluster 2. We have our inorganic anions in this cluster 2. We have the trifluoromethane sulfonic acid in the cluster 2. So we have some of our highest concentration chemicals we found only in a few samples, so they're not that widely spread apparently. But when they're present in high concentrations. Based on this data, we wanted to see, or at least have a first indicator if this is relevant for human health. What we did, we calculated human exposure. We calculated it based on a worst case approach. So we took the 95th percentile concentration in our data set, assumed 2 liters of tap water consumption a day, and an average weight of 70 kilograms. With this, we calculated this human exposure you can see in the graph, and we compared it to so-called Kramer classes. What is a Kramer class? In the end, it's a structure-based classification of chemicals. And based on the structure, the chemicals get assigned a Kramer class. And these Kramer classes have estimated toxicity values. With this, you can calculate the threshold dose. Kramer class 3 was the class where most of our chemicals fell into, and it's also the class with the lowest threshold. It has a threshold of 1.5 micrograms per kilogram a day. And all of our chemicals were at least one order of magnitude below this threshold value. Of course, there are several things to consider. Only a part of the human exposure is through drinking water, so it should only account for roughly 20% of human exposure. And this Kramer class is really an estimate only. It's based on the structure. It's not based on toxicity data for these chemicals. So they could be much less toxic, or in some cases, maybe more toxic. And if you have a look, among our highest human exposure chemicals, 3 of them could not be assigned to Kramer class by the tox-free software we used because they were structurally too different. This indicates that at least on first glance, with this rough estimation approach, there doesn't seem to be a critical human exposure, but there are many uncertainties that have to be taken into account, like toxicity of individual chemicals and the effect of the chemicals that cannot be assigned a class, and of course, cumulative human exposure through different sources. Looking at this whole pathway from wastewater treatment plant over bank filtration to drinking water, there are some conclusions we have to draw. First of all, bank filtration and even advanced water treatment is only partially effective against PM chemicals. We saw removal in every step, but we also see the same chemicals popping up again and again because they can break through these barriers and they are some of the high concentration chemicals. We saw that source water quality seems to have a large effect on tap water concentrations. And this leads us to one main conclusion, at least from my perspective, and this is that drinking water protection begins at the source. We have to take actions at the source against those most critical PM chemicals for which there's no effective removal technology if they prove to have adverse health effects. This concludes the field studies I wanted to talk to with you, but we also had a lab study we did in a project where we wanted to assess what happens if you use wastewater for agriculture. What effect do PM chemicals in this water have? Is there plant uptake? So is this another human exposure route? and can there be infiltration into groundwater? To do so, we did two studies. One of them is already finished, and I will focus on this one, where we used rocket and planted in a greenhouse and irrigated it with spiked fresh water to 80% field capacity. Here we wanted to find out which of these PM chemicals are taken up by plant and if they are transported to the edible parts or not. And another study, this is currently still in progress, we're currently still evaluating the data. Here I only give you a quick sneak peek, where we want to find out with soil column studies which of these PM chemicals actually have a high likelihood of reaching groundwater. Let's have a look at the plant uptake first. When you irrigate plants with water-containing chemicals, there are several factors that may influence the concentration in the plant ultimately. You have, of course, sorption to soil, which is likely less relevant for many of the PM chemicals we are investigating. We have always the question how much of the water containing the chemicals is actually taken up by the plant, which can hugely vary with experimental time. And we have the potential of elimination of chemicals, either through volatilization, which is also less likely in this study because none of the chemicals we use is especially volatile. And there's, of course, potential for transformation in a plant. Of these factors, we found this amount of water taken up likely leads to the largest variance between different studies, because this can very much depend on the growth period. It can very much depend how long is the experiment running. So we decided to closely monitor the water taken up by the plant. We had non-planted pots in which we estimated evaporation. We closely monitored the irrigation and we analyzed how much water remained in the soil after the experiment. And with this, we calculate for each individual plant how much water was taken up over the experiment period. With this information, we calculate then what we call the relative uptake. This is the total amount of chemical we find in the plant at the end of the experiment, divided by the total amount of chemical in the water the plant has taken up. So the total amount of water multiplied by the concentration in the water. So how does this look like? What you see here are the relative uptake values for the chemicals we investigated. First of all, this is a kind of metric you don't really see often. So the first thing we need is an anchor point. Therefore, we look at carbamazepine. This is a chemical very frequently studied for plant uptake. It has been studied in all kinds of different plants, and it's usually among the highest, if not the highest plant uptake chemicals in many studies. If you have a look at our data, 44 of the chemicals we detected have a relative uptake below 20%. And only roughly 10 chemicals have a relative uptake higher. So we again have the picture here that some chemicals show exceptional behavior. What was quite remarkable for me, that we have seven chemicals with a relative uptake higher than that of carbamazepine, which is usually the top end or near the top end in many studies. If we have a look at these chemicals, we again have our two inorganic anions popping up. We have one pharmaceutical with oxypurinol, and we have four short-chain PFAS. We have the widely known free fluoroacetic acid that you can, by this point, find pretty much everywhere in the environment. We have free fluoropropionic acid, which is just one chain longer. We have the free fluoromethane sulfonic acid. We also found high concentrations in tap water. And again, we have this bistriflamide, this novel PFAS that's often connected to energy storage because it's used in lithium-ion batteries. So with some of these chemicals taking up exceptionally well, the next question for us was, do they remain in the roots or are they transported to edible parts of rocket? Therefore, we calculate translocation factors, which is basically just a concentration ratio between the chemicals in the roots and the chemicals in the leaves of the rocket. Translocation factor greater 1 means we have enrichment in the leaves. The chemicals are predominantly transported towards leaves. and below 1 means they mainly remain in the roots. We have around 2/3 of our chemicals with a translocation factor greater than 1. Again, we have the carbamazepine as our anchor point with a translocation factor of 23. So you see it's very effectively transported into the leaves. Our PFAS and inorganic anions all show transportation to the leaves as well. Some of them very strongly with translocation factors between 5 up to 43. And our oxypurinol is also mainly transported to the leaves with a translocation factor of 3. What was quite interesting here, that we have 3 either guanidines with cyanoguanidine and the DPG, or a chemically similar compound with the acetoguanamine with very high translocation factors. So these chemicals seem to be very effectively transported to the leaves of rocket. From this data, we can learn that our high uptake chemicals don't remain in the roots, but they are also effectively transported to the leaves, so to the edible parts of rocket. We wanted to compare our data to literature. Since the RU is not right reduced, we calculated bioaccumulation factor, bioconcentration factors. Since we have low sorption to soil, you often use soil concentrations to plant concentrations. We decided to compare it to hydroponic studies and calculate a biotransformation factor from water to plant, because otherwise this would be quite pointless exercise for chemicals that do not absorb to the soil, because it only depends on how wet the soil is at this point. To make it somewhat comparable, we only looked at experiments in literature that use similar plants, so rocket, lettuce, spinach, and fairly long cultivation times, 21 to 55 days. If you look at the figure A here, you see the bioconcentration factors. Bioconcentration factor in our study was higher. It was somewhat statistically significant with a p-value below 0.05, but this can at least partially be explained by a longer experimental time. This again highlights the use of RU that really accounts for the experimental time, that accounts for the water uptake. So this difference in the bioconcentration factor is somewhat significant, but not too astonishing. What was much more interesting was the much stronger translocation to the edible parts of the plant, to the leaves of the rocket in this case. Our very polar chemicals from this study were by an order of magnitude stronger transported to the leaves than less polar chemicals from literature. So it indicates that while very polar chemicals might not be taken up by plants much stronger, they are much more readily transported to edible parts apparently. Of course, we can only speak for leafy greens right now. For plants like tomatoes, this would need additional studies. Another thing I want to point out is that we observed high uptake for perfluorocarboxylic acid, especially for the short chain ones. This is in line with literature and quite expected. What was unexpected was this remarkably high uptake for TFMSA, because usually the fluorinated sulfonic acids show fairly low plant uptake. It was the same in our study. They showed very low plant uptake until we reached the TFMSA, where the plant uptake suddenly skyrocketed, which means maybe at this very short range, different mechanisms play a role. Finally, after we looked at the plant uptake, I want to give you a first sneak peek in our study for the infiltration. What you see here is the retardation coefficient. So we used the tracer bromide and calculated how much faster was the tracer leaves the column than our chemicals. And basically built 4 categories out of this. Category 1, the green ones are tracer-like. So they reached the end of the column with the tracer only shortly after. Then the categories 2 and 3 are somewhere in between. And the category 4 did not leave the column during the experimental time. What you can see here, we have more or less a split half and half. We have a small section in the middle of chemicals that leave the column but are somewhat retained. And then we have 2 majorities of chemicals, one that does not leave the column. Either they are strongly sorbed there or they undergo transformation. And one that leaves the column really quickly. together with a bromide tracer. From these results, we can draw some conclusions for agricultural wastewater reuse. The first thing is that this relative uptake seems quite effective to increase comparability between studies. We see that some PM chemicals are taken up readily by rocket and are rich in its edible parts. This was especially true for ultra-short chain PFAS and those two inorganic anions we have. And lastly, that some PM chemicals show tracer-like behavior, showing that there's a high likelihood to reach groundwater. From this, we conclude that it's important to include key PM chemicals into risk assessment and quality control for agricultural wastewater reuse. With this, we finished our way through the water cycle. But now I want to have a closer look what we can learn by comparing these studies to one another. We saw this plant uptake, but of course, a chemical that's taken up well doesn't mean it's present in high concentrations. It doesn't mean it has a high human exposure, because it also depends on the concentrations present. So we took the study from Muschke et al., who also had a look at several German wastewater treatment plants and plotted the median concentration in the wastewater treatment plant against the relative uptake. And you see the chemicals on the top right, they combine high wastewater concentration with high plant uptake, and we call this the human exposure potential. So they have a high potential for human exposure in agricultural wastewater reuse. We see on the very right side, we see our high uptake chemicals, BF4, PF6, carbamazepine with high wastewater concentrations. And then below you can see the perfluoropropionic acid, TFMSA, and TF2. They all had a high plant uptake, but were present in much lower concentrations in the wastewater. In this wastewater study, TFA was not present, but if it wouldn't, you would assume general TFA concentrations in wastewater, it would also be at the top right side. Of course, this is only based on exposure potential, so chemicals that are toxic or may have hotspots might have a high priority as well. We then try to transfer the same approach to groundwater. Here we took the relative amount in the effluent of our column, so how much of what we added left the column in our experimental time, and plotted against the same wastewater concentrations. And you can see some of the same chemicals popping up at the top like the PF4. If you then start to plot both against each other, the human exposure and the groundwater exposure potential, you can see which chemicals may be very critical for agricultural wastewater reuse. And no surprise, this BF4 shows up at the very top right because it has a very high potential to reach groundwater. It has a very high potential to reach humans. So we can see here that PM chemicals pose a challenge to agricultural wastewater reuse. And they pose it not only for human exposure, they also pose it for groundwater contamination. So we have to have a closer look here. What's the next thing we saw? We saw that there is quite a variation in PM concentration and PM presence in different samples. In our two wastewater treatment plants, we saw that quite a few chemicals were only present in one of them, including high concentration chemicals. In our two river systems, we observed the same thing. We had a lot of chemicals that were present in both in comparable concentrations, but roughly 1/4 of our chemicals were only present in one of the river systems. And this also includes some of the high concentration chemicals. And in our tap water, we also saw we have those less frequently detected chemicals that contain some of the highest concentrations. So local sources seem to be very relevant for PM chemicals and for water quality. So we have to have a closer look. The problem with these less frequently detected chemicals, they might easily escape screening approaches if you don't have the right samples. Another thing that's quite important to me is if you have a look in our barriers, at some point they all rely on transformation as a major mechanism. We saw it in the wastewater treatment. The transformation can lead to formation, even though we didn't intentionally include transformation products here. In the same wastewater treatment plant with the ozone treatment and the activated carbon, We also had a non-target screening. I don't want to go into too much detail here, but every dot represents is a feature, so something we detected, a pair of a mass and a retention time. The purple and the red dots are chemicals formed during ozonation. So those are transformation products that are newly formed. And the red dots are those products formed during ozonation that are not removed by subsequent filtration through activated carbon. So you see there's a huge potential to form mobile transformation products that is so far rarely accounted for. And this is, I think, still a gap we have in chemicals management and need to address more. And the final thing I want to talk about is there were a couple chemicals that popped up again and again and again throughout the water cycle we discussed today that showed poor removal during advanced water treatment, poor removal during bank filtration, strong uptake to plant and translocation to edible parts. We had a high potential to reach groundwater. They were present in drinking water. And almost all of them fall into this chemical space of small, very persistent anions. We have, of course, our inorganic chemicals here and the PFAS. Some of them, like TFA and the propionic acid, are known for a long time. But some of them we discovered fairly recently in the last 5 to 10 years. So this is a chemical space that is worth further investigation. Those small, very persistent anions, because here we seem to have the highest potential to combine negative effects, negative properties of chemicals. With this, I come to an end. Due to the time, I could only give you a short overview. If you want a deeper dive in these topics, I recommend the following papers. And in the end, I want to thank the people that did most of the work. The first authors of three of the studies I presented today, Isabel Neuwald, Matthias Muschke, and Alina Selig. Of course, all the co-authors that contributed to this work, and the project partners, and the BMBF for funding the projects Protect and Poor, in which the work I showed you today was performed in. With this, I'm done, and I'm looking forward to your questions.
Many thanks, Daniel. That was an excellent presentation and really enjoyed going through this work that you've done recently and how you kind of connected the entire water cycle. I'm just going to remind everybody that we have 12 minutes for a Q&A. So if you have any questions to Daniel, please put them in the chat and I will ask them. And I also have some of my own questions. And just to introduce myself, I'm Hans Peter from the Norwegian Geotechnical Institute. So yeah, the first is from an anonymous attendee. Our first question, they ask, is there any chemicals excluded from the study due to the source being pesticides such as TFA? But I think maybe you could also answer that by saying, like, how do you choose what chemicals that you decide to include in your studies?
It was mainly based on work we did at the beginning of the Protect project. In 2021, we did suspect and non-target screening in different waters, mostly surface water. And based on the results there, we prepared the list for the following studies.
Mm-hmm. Yeah, so just to specifically answer that question, that was the basis. Like if they're from transformation of pesticides, that was
We did not
independent.
consider this, no.
No, and I recall it's mainly had an industrial chemical focus. Like
We had an industrial chemical focus. We took different lists of supposed PM chemicals, of course, as starting point for our screening approach, including the one of Uber.
mhm. Great, thank you. And so next question: do we know more about the toxicity of N2F2? Is it a byproduct or used as such for battery production?
It is used as ion in the electrolyte of batteries, but of course the batteries alone cannot explain this widespread occurrence of this chemical. We find it in low concentrations, but we find it pretty much everywhere. Of course, it's known to have hotspots that are near battery production, battery recycling, but I don't think batteries alone are enough to explain this widespread occurrence. So there must be other sources we don't really know of or don't know of yet. And this chemical is quite interesting because it combines so many detrimental effects. It's very persistent. It's not destroyed by any treatment technology, even if you do a top assay, which is very strong oxidative conditions, this chemical remains untransformed. It can reach groundwater. It's readily taken up by plants. It makes its way to drinking water. It's not really removed. What we don't know much of is the toxicity. But other than that, it combines pretty much every negative effect the chemical can have. So it has all it needs to spread quickly in the water cycle. The question that remains is how it's its effect.
Mhm. Yes, and the next question is related. So the focus on public debate currently is, oh, and I should have said, yeah, I was Joanna Berda that asked that previous question on NTF2. The next question is a anonymous question. The focus on the public debate is mostly on TFA. When it comes to very persistent anions, PFAS, do you know about the toxic properties of the other ones that you showed that have the highest potential of negative effects? So maybe just extend your previous answer to other anions.
They are not well investigated yet. The TFA is the one where you know the most. We have now very recently this reclassification that was proposed from Germany that it has reprotoxic effects. But of course, the others are not that well studied yet. For TFMSA, there was a study that said it has adverse effects in the on the gut microbiome of mice, but there is not much more that I'm aware of.
Mm. And well, one thing I think that was quite interesting with your results also, and I think it should probably be a thing that we look more into, is the accumulation of these small ionic substances in plants, because this could be a huge part of the exposure to these chemicals. The focus, for instance, of TFA has been on water exposure, which can be quite substantial in some areas. But the numbers that we're seeing coming in from the plant uptake studies are huge, and so there haven't been any good estimates on diet from plants versus water. But what is your feeling about, based on your work with Rocket? Are you more? Do you think? What is your suspicion? Like to explore and further research, are plants and vegetables a higher source of these chemicals than water?
They can be fairly relevant for several reasons. Of course, we observed this really high uptake. It was in rocket, it's a leafy green. Of course, we have to have a look in other plants like tomatoes, etc., if it's also accumulating there. But of course, there is a high potential for these plants to be exposed to TFA. TFA is in rain, so it can be exposed to rain. It's also a metabolite of quite some pesticides. So there are many ways that the plants can be exposed to high levels of TFA.
Mhm.
So this could be a substantial human exposure pathway.
Mhm. Great. And this next question comes from Evan Bolton from PubChem and others. Exploratory question. With so many concerning PM chemicals only recently discovered, what else is PM in the water that we are not looking for? Example, that different columns or approaches might be able to separate out. So what are the next ones you're going to be looking for?
I think in my last slide, I tried to point out a bit that this chemical space of the small persistent anions might be especially interesting because the chemicals we found there seem to have those negative effects of being able to spread very effectively, not being removed, accumulate into plants. This is one avenue we have to look at. And the other one is this issue with transformation products. We have some chemicals that may be formed from many, many precursors. So usually the idea is when you have a chemical, you release it in the environment. Transformation products are not so relevant because many transformation products are formed from one precursor. Concentrations get lower. This is often an argument why transformation products are not so relevant. But of
Mhm.
course, you can have it the other way around, especially when you have these small, very basic molecules that may be formed. You can have many transformation products, one transformation product or few transformation products formed from many precursors. There was a very nice presentation at CTEC from Emma Palm about this.
Emma Palm and 0 PM, I should
Yeah,
have.
that showed that especially for those very simple molecules, you can have many precursors. So concentrations of individual transformation products can be much higher than you would expect.
Mhm. Yeah.
This is another way to go. And I think one that's still difficult to tackle and where we are still exploring what's the best way to tackle this.
Yes, absolutely. And it's also important to point out that a lot of transformation products are negative, right? Because it goes through oxidation, which can form things like carboxylic acids. Next question from Rita Benetti. Thank you for your excellent work and clear presentation. A lot of attention is given to the quality of purified wastewater for reuse in agriculture, but as also partly highlighted during your presentation, less is known about the quality of water of both groundwater and especially surface water. Do you think this approach, I mean, risk assessment, like you did, should be applied to any type of water use in agriculture?
I did it with wastewater here because the project was based on agricultural wastewater reuse in which we did it. But if you have a close look, one of the chemicals that was near the top for this human exposure potential for wastewater reuse was again the BF4, which is also a chemical that's present in very high concentrations in surface water. So even if you use surface water for irrigation, I would not expect the human exposure potential to be much lower. Because also in surface water, we saw concentrations up to 2.6 microgram per liter. So some of these high concentration PM chemicals, they can also be agriculture can also be a significant source of exposure even if you don't use wastewater, if you use surface water. We also had this example with TFA. Using wastewater would probably not have that much an influence on the TFA concentration because a lot of it is also from rain, a lot of it is from metabolites, from plant protection products.
Mm-hmm.
So there are selected chemicals for which irrigation with surface water won't make a large difference.
Mm-hmm. Thanks. And suddenly we got an explosion of questions in the Q&A. So I don't think we'll be able to answer all of them in the next minute and a half. Some of them are from anonymous attendees, but what we will do is if there's a name next to your question, we will send it to Daniel and see if he's got time to answer it. If you want to increase your chances of that, please repost your question, not as anonymous, but the actual person, so we can get in contact with you, or Daniel can with his answer. And we got a question just going down the list from Michael Neumann. Thanks for the great overview and your conclusion at the end. How to regulate inorganic small anions as they are excluded from the new vPvM hazard class under CLP? Any ideas? I mean, how do we regulate? I know you're not a regulator, but did you think about that?
This is a question I actually would want to reflect back to do, as you're a regulator.
Michael, we should ask you that.
I don't know what's a good way to address these chemicals. We saw those two inorganic ones that really pop up again and again because they have high concentrations. Of course, most what we look at are organic chemicals because we expect a higher potential to be toxic for them. For these two inorganic chemicals, we actually don't really know. The problem is those ionic liquids are often tested as salts. So it can be the toxicity there is mainly dominated by the cation, but we are not 100% sure.
Mm-hmm.
So for these chemicals, we just need to answer the question about toxicity and adverse effects, I think.
Mm-hmm. Okay.
But in general, we should avoid high concentration chemicals in our plants and in our tap water.
No, I think that's very good. And I'm looking at the time, and there's some very good questions from Evan Bolton. Thanks. We'll get back to you on that. And from Michael Bernholm, we're out of time, but so I think we've tried to end these on time. But I want to thank you, Daniel, for your excellent presentation. It stirred up a lot of interesting conversation, and your results are extremely important as we go forward on also our project towards zero pollution of persistent mobile substances. So I'd like to thank you again, and I'd like to thank the audience for your participation. And if you're interested in webinars like this, please follow us on LinkedIn where we announce this. And I'm also going to highlight we have a special workshop in Greece in October 7th to 8th on removal of persistent mobile substances. So if you're interested in talking about how to remove these from wastewater, from drinking water, surface water, this is the workshop you need to go to. It's in Greece. More info is on our LinkedIn and websites. And with that, we'll close this webinar. So I'd like to thank you on behalf of 0PM from me, Hans-Peter Arp, and from Sarah Hale. And thanks again, Daniel. Goodbye.
Thanks for having me.
0PM. Zero pollution of persistent and mobile substances. This project has received funding from the European Union's Horizon 2020 Research and Innovation Programme under grant agreement number 101036756.